Hi everyone, this video is going to cover muscle structure. A muscle is an organ, and the organ is made up of bundles of fascicles. Those fascicles are made up of bundles of muscle fibers, and muscle fibers is just another term for the muscle cell. The muscle fibers are made of bundles of myofibrils, and the myofibrils are broken down into small segments called sarcomeres. And the sarcomeres are the contractile units. That's where the contraction occurs. And within the sarcomeres are myofilaments. Here is a nice illustration of a cross-section of a muscle. Muscle attaches to bone through dense regular connective tissue. If it attaches in a rope-like structure, the rope is called a tendon. If it attaches in more of a sheet, the sheet is called an aponeurosis. Surrounding the muscle is connective tissue that's called epimysium. The epimysium, again, is the covering of the organ. Surrounding the fascicle is the perimysium, and surrounding the muscle fiber within the fascicle is the endomysium. So each muscle fiber has its own connective tissue that surrounds it. Each fascicle has its own connective tissue that surrounds it, and each muscle has a sheet of connective tissue that surrounds it. A muscle fiber is a highly adapted cell. It is very long. In fact, most muscle fibers go the entire length of the muscle that they are part of. It has a specialized cell membrane called the sarcolemma. It has multiple nuclei. It is made of many myofibrils, kind of bundled together. And you can see that the myofibrils are broken down into little segments, and those segments are called sarcomeres. So just kind of keep that in the back of your head. We'll come back to that shortly. And a muscle cell has a lot of mitochondria because it needs a lot of ATP. Here we're going to look deeper into a muscle fiber. I mentioned in the previous slide the sarcomere, and this is kind of a detailed illustration of a set of sarcomeres. Here we can see that the sarcolemma actually folds in to the muscle fiber. Now the sarcolemma is the cell membrane, and this cell membrane is similar to the cell membrane of a neuron in that it can carry an action potential. It's a slightly different action potential than what we saw in neurons, but there's many similarities. When that action potential reaches this area, called the T-tubule, the action potential is carried down into the muscle fiber. The T-tubule is surrounded by this region right here, which is actually part of the endoplasmic reticulum. And in a muscle cell, the endoplasmic reticulum is called sarcoplasmic reticulum. It forms this blue network that you see surrounding the myofibrils. At the edges of the blue network, along the T-tubules, you see it gets wider, and those regions are called the terminal cisternae. The terminal cisternae is where calcium ions are released in response to an action potential being sent down the T-tubules. The terminal cisternae and the T-tubule forms a triad. Another thing we should look at in this illustration are the details of a sarcomere. We've seen in muscle cell slides that muscle cells are striated. They have light and dark stripes. Those light and dark stripes are formed by the I bands over here and the A band here. In the center of the I bands, we have an area called the Z disc that zigzags like a Z. The Z disc forms the border of a sarcomere. So from Z disc to Z disc is a single sarcomere. Within a sarcomere, there are what's called thin and thick filaments. The thin filaments are made of actin, and the thick filaments are made of myosin. And in lecture, we'll go over the way that actin and myosin work together to shorten a sarcomere. What I'd like you to notice right now is that from the Z disc going in, we can see the thin actin filaments. And in the middle of the A band, along the M line going out, we can see the thick filaments, thick myosin filaments. Just a few more details I want to get into about the A band. So where the M line is where those thick myosin filaments attach. But there's a small region called the H zone, and the H zone is where we just have thick filaments. The actin doesn't reach all the way into the H zone. Okay, and so this is ever so slightly lighter when you look at it in a microscope. And so just to tie this all together, we're going to go back to that neuromuscular junction that we've seen so often this semester, and now look at it from the muscle's point of view. So we're familiar with this axon terminal releasing neurotransmitters, and in this case it's going to be acetylcholine. Acetylcholine is the neurotransmitter for skeletal muscle, and the acetylcholine is going to bind to the receptor over here. And if a sufficient amount of acetylcholine binds to the motor end plate, to the receptors along the motor end plate, an action potential will be sent down that sarcolemma. So we find ourselves back to this neuromuscular junction model. Earlier in this semester, we were paying attention to the axon and the myelin sheath and the axon terminal. But now I would like you to pay attention to the parts of the muscle fiber. So this is a muscle fiber. This membrane right here is the sarcolemma, and the infoldings of the sarcolemma form the T-tubules. We can see the triads formed of the terminal cisternae and the T-tubule. And it's important to note that in this model, the sarcoplasmic reticulum is beige, and in the illustrations from our textbook, the sarcoplasmic reticulum is blue. So don't get confused there. There's a reverse coloring. The T-tubules in this model are blue. The T-tubules in the illustration were more of a whitish. But here we can see that sarcoplasmic reticulum ending in the terminal cisternae, T-tubule, and another terminal cisterna. So this is where we get that triad again. We can see the light and dark banding. So a sarcomere, remember that, so this is where those Z discs are. So a sarcomere is going to be from this Z disc to this Z disc. That forms a sarcomere. Okay? If this forms a sarcomere, what this tells me about what I'm seeing up here is that this is a myofibril. In between the myofibrils, you see many little red mitochondria. And we also can see an example of a purple nucleus. Now remember, muscle fibers have many nuclei, so this is just one. A side view of this model shows us the connective tissue that surrounds the muscle fiber. This is again called endomysium. This model shows the ultrastructure of a muscle cell. So here we have those myofibrils that are broken down into sarcomeres. So here's that Z disc in the middle of the I band. Here's the A band with the M line and the H zone. We can see the sarcoplasmic reticulum ending in the terminal cisterna, a T-tubule, and another terminal cisterna. Here, these are mitochondria. And you might be saying, well, I think that's actually a nucleus, but it's not. And the main reason that I know it's not is because A, the size of a sarcomere is much closer to the size of a mitochondria, and we see these infoldings, and these infoldings tell us that we're looking at the inside of a mitochondrion. I hope you found this video helpful. Remember to use both your lab manual, the most important points, and the practice anatomy lab tutorials on mastering to help you study for next week's quiz.